we'll start with kind of an overview of the nephron and an overview of the function of the kidney. So I'm going to do a bit of annotating here, so bear with me. Okay, so we're going to look here at an overview of the nephron. To start. And so I'm going to just draw out here loosely our nephron. Okay, we've got this afferent arteriole, this glomerulus, this efferent arteriole, and then we've got our capsule structure, right? So the Bowman's capsule. Okay, the proximal convoluted tubule. Got the loop of Henle here. Our DCT, or the distal convoluted tubule. And then finally, our collecting duct. Okay, and bear in mind, although I'm drawing this out here for simplicity, we want to have this 3D understanding that the distal convoluted tubule really wraps around and comes in contact with the afferent arteriole, right? There's this connection between the afferent arteriole and the distal tubular structures. So it's kind of more contorted than I'm drawing it out, but I am going to make it a little bit simplistic for our purposes, okay, for the purpose of explaining it. Okay, so let's kind of label these out here to start. So we've got this afferent arteriole, which is bringing blood towards the glomerulus. We've got our efferent branch of that, taking blood away. We've got the glomerulus here. I can't spell today. Okay, we've got the glomerulus here. We've got the proximal convoluted tubule, the distal convoluted tubule, and then our loop of Henle. Alrighty, and then the collecting duct. Okay, first thing we want to distinguish is that the glomerulus and this part of the tubular structure, so the PCT, DCT, this is all up in the cortex, okay? I'm gonna draw a line here, which is demarcating the cortex versus the medulla. Alrighty. Okay, so we're going to go through here part by part, and I'm going to speak about important things that we want to point out for each of these areas. So first thing is the glomerulus, right? Together, these two structures we described as the renal corpuscle, that is the glomerulus, and the Bowman's capsule. And let's talk about some of the things that are important here. First off, we know this region is in the cortex. That's kind of the first thing. We also want to point out the Starling's forces that are controlling filtration. So the Starling's forces. All right, I'm not going to go into detail with the Starling's forces, but make sure you understand the 4 different forces, what's going in what direction. And if you have questions on that, I can address those separately. But here we're kind of looking at an overview. I got a question when I'm going to post exam. So I'll talk all about the exam towards the end of this. All right, so stay tuned to that. So make sure you have an understanding of the 4 Starling forces, be able to explain them individually. So a good idea would be to kind of bullet point them. So talk about the hydrostatic, Bowman's capsule pressure, the hydrostatic capillary, glomerular capillary pressure, and so on, and be able to describe what direction the fluid is moving according to that force, whether it's a push or pull, etc. Other important things here is understanding, this is where glomerular filtration takes place. So the process of filtering out that plasma, you should be able to explain what goes through the filtrate, right? The fact that proteins and RBCs do not, they're too large, they stay behind, and what ends up in the tubular space that becomes our tubular filtrate. Also, we talked here about intrinsic regulation. Okay, and the three processes here were the myogenic feedback, the tubuloglomerular feedback, and then the response elicited by the mesangial cells. Okay, also have a clear understanding of these three mechanisms and what they're doing. Okay, in brackets here, I'm going to put GFR maintained at 125. Okay, we know that that is the function of the intrinsic regulation, right? It's going to keep GFR maintained at 125 milliliters per minute as long as our mean arterial pressure is between 80 to 180. Okay, actually, I'll go ahead and put that here. So if mean arterial pressure is 80 to 180, then GFR is maintained at 125. So that's the intrinsic part of this. The extrinsic mechanism we talked about as being the regulation that happens if mean arterial pressure goes outside of that window. So if mean arterial pressure goes below 80, so let's write that here. Mean arterial pressure is less than 80 or greater than 180, then GFR will either rise or fall appropriately, okay? Depending on which direction, and that is elicited by the sympathetic nervous system. Okay. So that is the mechanism for the extrinsic regulation, and then we talked about three mechanisms for the intrinsic regulation. So have a good understanding of how those differ, how those different mechanisms are brought about. Right, and I think that's pretty much it for the glomerulus and this first part of the nephron. Let's talk about the proximal convoluted tubule. So as far as the PCT, let's talk about some of the structural differences here. First off, we know that the cells here have leaky tight junctions. Okay, meaning that there is this ability for paracellular transport, so things can leak in between the sides of those cells. There's also microvilli, which creates the brush border. And that is a mechanism or an adaptation, which this should be brush, sorry. Brush border. So this is an adaptation which increases the surface area of the proximal convoluted tubule and increases or enhances reabsorption, okay? So here we see unregulated reabsorption. Meaning that it's happening absent of any stimuli or any hormone. It's happening freely. And this is happening according to that solute reabsorption, which then facilitates water reabsorption, okay? Alrighty. Other things that are reabsorbed here, we know amino acids, glucose, right? 100% of the glucose that is filtered out. We talk about bicarb being filtered here, reabsorbed here, excuse me, as a part of our acid-base balance. Bicarb, glucose, water, amino acids. Yeah, so those are the most important things that are reabsorbed in the proximal convoluted tubule. This reabsorption happens unregulated according to the leaky tight junctions and the enhancement that is brought about by that microvilli, that brush border. I also want to make a note here about the peritubular capillaries. So there are capillaries that are wrapping around the proximal and distal convoluted tubules. We call these peritubular capillaries. And this is where those reabsorbed substances are moving to, from the lumen of the tubule into these peritubular capillaries, okay? You also want to understand the barriers to reabsorption and secretion. So the barriers that these substances must move across, and we're going to talk about that as a part of one of the essay responses. So I won't really put it here. Actually, let's move this. There. Okay. Moving on, let's talk about the loop of Henle a little bit. So the loop of Henle was this part of the tubule that dips down into the medulla. Here is where we saw the medullary osmotic gradient. Which was brought about by the countercurrent multiplier. Okay, so make sure you understand what that means, right? The gradient, which is the fact that from the cortex down into the medulla, we have this gradient of osmolarity, not only in the medullary interstitial fluid, but also in the filtrate. Okay, the osmolarity of the filtrate gets higher as we go down. So does the osmolarity of the medullary interstitial fluid. It gets higher as we go down into the medulla, and as we go back up towards the cortex, that osmolarity is higher as it gets back to that 300, which is what the cortical fluid is at, 300 milliosmoles. So have a good understanding of that, have a good understanding of how it's brought about, the fact that the descending limb, permeable to water only, and the ascending limb, permeable to solutes only. When we say solutes, we're talking about sodium, potassium, and chloride. Okay. And so that sequence of events that is brought about by those two differing permeabilities is what brings about that gradient. So have a good understanding of that. And again, I'll take questions specific to that if there are any. We also want to point out here that the vasa recta is intimately connected to the loop of Henle. This is where those substances are being reabsorbed to. So we want to talk about the fact that the vasa recta has an opposite orientation to the direction of flow in the tubules. So as we go in this direction in the tubules, descending to ascending, in the vasa recta, we're going in the opposite. So the ascending limb of the vasa recta is in contact with the descending limb of the loop of Henle and vice versa. So have an understanding of that. Also, the role of urea. Okay, the role of urea in maintaining that gradient. Yeah, and I think that's it as far as the loop of Henle. Let's bring this out a little bit. Okay, now we speak about the distal convoluted tubule and the collecting duct pretty much as one. The features here overlap. So we'll talk about them.
Oh,
Is there a question?
I'm in class.
Okay, just make sure your mic is on mute. I think I hear a bit of a background conversation. Okay, so let's speak about the DCT and the collecting duct together. Again, features here kind of overlap. Here we have regulated reabsorption. We see regulated reabsorption of our solutes. So that's going to be sodium, potassium, chloride, and water.
Oh, wow. This wasn't reviewed for my exam Friday. I'd talk more, but.
Okay. So that's going to be regulated. And when we say regulated here, this is happening because the cells here have tight junctions, meaning that there is no paracellular transport in between the sides of these cells because of these tight or very secure tight junctions that are seen here. And then we also have the influence of a few hormones. Oh wait, we forgot to talk about, so we have regulated absorption of solutes. We also have secretion here. Okay, let's not skip over that. So we do have secretion of some solutes, mainly potassium, and hydrogen ions, okay? So remember that secretion of hydrogen ions here is a part of our acid-base balance that is regulated via the kidneys. The reabsorption of bicarb in the proximal tubule and the secretion of hydrogen ions in the distal part of the tubule is how we kind of regulate that acid-base balance through the kidneys. So we do have a bit of secretion here, unlike the PCT where we have zero secretion. And then we also want to talk about the hormones that influence reabsorption here. Because reabsorption is regulated, it's going to be under the influence of hormones, ADH for one, right? Aldosterone. And remember, aldosterone is that final product in the renin, angiotensin, aldosterone pathway. So make sure you're making that connection here as far as aldosterone is concerned. And then ANP, atrial natriuretic peptide. Okay, let's actually describe these here a little bit further. ADH, we said, is going to enhance water reabsorption. So that's the hormone that influences water reabsorption mainly, but we know that sodium is also going to be influenced here as well. But the main role of ADH is water reabsorption. The mechanism is going to be through aquaporin 2 channels. Aldosterone, we said, the main mechanism is sodium reabsorption. And the way that aldosterone does that, we spoke about two things that aldosterone does. It's going to increase the sodium-potassium pumps on the basolateral membrane. And it's also going to increase the leak channels for sodium on the apical membrane. Okay. Right, so those are the two mechanisms by which aldosterone increases sodium reabsorption in these distal parts of the tubule. And then lastly, ANP we know has the opposite effect. It is going to decrease sodium reabsorption. I'm going to put increase here just so there's no confusion. So we're increasing sodium reabsorption via aldosterone, and we are decreasing sodium reabsorption via ANP. And the way that we do that is by blocking the leak channels on the apical membrane only. Okay, so these are the three hormones that influence the reabsorption taking place in the distal convoluted tubule and the collecting duct. And these are how these three hormones regulate the reabsorption of water, sodium, water and sodium. Let's talk about how aldosterone also brings about the secretion. So I'm going to bring it here. Of potassium, okay? So aldosterone increases sodium reabsorption, but it's going to decrease or increase rather potassium secretion. And it's this exact same mechanism. Remember, the sodium potassium pump is pumping potassium into the tubular cell, sodium out of the tubular cell. So if we increase aldosterone, we're going to put more of these sodium potassium pumps in the basolateral membrane. That is going to bring more potassium into our cell, and then the leak channels is going to bring more potassium into the tubular fluid. So aldosterone is having two effects by these two mechanisms. That is going to be increasing sodium reabsorption, but also increasing potassium secretion by putting more basolateral sodium potassium pumps and more apical leak channels. That's going to facilitate both of those effects. Okay. Let's see if there are any questions so far. Okay, I'm not seeing any questions there. Yeah, and I think that that is all that we spoke about in terms of the different parts of the tubule and the different functions. Okay, I can't think of anything else that we maybe overlooked. All right, and I don't see any questions. I'm going to move right along. I'm going to save this. I'll upload this to Blackboard for the benefit of those who may not be tuned in right now. Okay, so let's clear